Here is the disclosure slide. We will have a total of 3 lectures on this topic, the autonomic nervous system drugs. Today, the first lecture will be focusing on the anatomy and physiology of the autonomic nervous system. We will also talk about the receptors and the intracellular signaling pathways that are activated by these receptors for this system. We will also talk about the mechanisms of the neurotransmission. Although today we are not gonna talk about the drugs, but today's lecture is the foundation to understand how the drugs work for the following 2 lectures. In the second lecture, we will learn about the drugs for the sympathetic nervous system. And in the third lecture, we will talk about the drugs for the parasympathetic nervous system. For the exam, for the second midterm exam, the questions will be based on the slides that I showed during the lecture. We know that the nervous system controls the human body, and the central nervous system, or the CNS, is composed by this brain and also the spinal cord. The function of the CNS is to integrate the information received from both inside and outside the human body to make decisions, and then to coordinate the activities of the different parts of the human body. The peripheral nervous system, or the PNS, refers to these nerve fibers that connect the brain and spinal cord to the different parts of the human body, and they are composed of these the fibers that are originated from either the brain or from the spinal cord. The peripheral nervous system is further divided to the somatic nervous system and the autonomic nervous system, or the ANS for short. The somatic nervous system refers to the somatic nerves, or they are also called the motor neurons, as shown here. They control the contraction of the skeletal muscles in the body for the voluntary actions which require conscious thought. For example, when we throw a ball, the contraction of the skeletal muscles in the arm is controlled by these motor neurons. Because it is a voluntary action, we can decide when and in which direction we throw the ball. So it's under a conscious thought control. In this figure here shows a segment of the spinal cord, and here shows the skeletal muscle, and here shows the individual cells of the skeletal muscle. We can see the spinal cord is connected with the skeletal muscle cells by these motor neurons, and the cell body of the motor neuron is located in the spinal cord, and the fibers reaches out to form these neuromuscular junctions with the muscle cells. And when the neuron cells get excited, it sends the action potentials to the terminal of the neurofibers to release the neurotransmitter to trigger the contraction of the skeletal muscle cells. By contrast, the autonomic nervous system controls the involuntary actions, which are not controlled consciously. For example, the heart rate and the heart muscle contractility, which mean how fast and how strong the heart contract, cannot be controlled consciously. These are the involuntary actions of the heart. Also, the contraction of the smooth muscles in the body is also not controlled consciously. The contraction of the smooth muscle in the blood vessel regulates the blood pressure. If the contraction is too strong, it may cause hypertension. The contraction of the smooth muscle in the lung airway controls the diameter of the airway tubes, which regulates the resistance to the airflow and to the ventilation of the lungs. For example, during exercise, the lung airway will dilate so that it will be easier to breathe in more air during the exercise. The contraction of the smooth muscle in the gastrointestinal tract or the GI tract regulates the mobility of the tract, which is important for the physical digestion of the food inside the GI tract. In the eyes, the pupil size is also controlled by the smooth muscle in the iris muscle. For example, when we stay in a dark room, the pupil will dilate to allow a maximum amount of the light to enter the eye. But when we turn on the lights, the pupil will constrict and become smaller spontaneously. So there's it is involuntary action. It's a spontaneous response of the eyes. The smooth muscle in the urinary bladder wall also controls the urination. When the smooth muscle contracts, it squeezes the bladder to facilitate the urination. The gland secretion in the human body is also controlled by the autonomic nervous system. For example, the lacrimation, which means tear secretion that occurs when people get emotional, and salivation when people eat a meal, and the secretion in the GI tract is also controlled by the autonomic nervous system. And this secretion is important for the chemical digestion of the food. The sweat glands on the skin are also controlled by the autonomic nervous system, and they regulate the body temperature. These are the some examples of the functions controlled by this autonomic nervous system in the body. This system is further divided into two opposing systems. The first is the sympathetic nervous system, and the second is the parasympathetic nervous system. Here on the right side, as shown here, it shows the sympathetic nervous system. These are the fibers in this system. Here we can see the fibers originate from this from the spinal cord, from these thoracic segments from T1 all the way down to T12. And it also originates from the lumbar segments from lumbar 1 to lumbar 3. These fibers reaches this structure called ganglia. And in the ganglia, they connect with a second neuron, as shown by the red color. And the fibers of the second neuron reaches the target organs in this system. We can see that in the sympathetic system, the first neuron, which is called the preganglionic fiber, is shorter, but the second neuron, the postganglionic fiber, is longer in this system. On the left side, it is the parasympathetic nervous system. Here we can see these fibers originate from the brain stem and also from the sacral segments of the spinal cord. Here I put X here because the drawing here is not very accurate. Here it should be from the segment 2 to 4, not from the first segment. We can also see that the first fiber, the first neuron, the fiber is longer. And the ganglia where the two neurons connect, they are located either near or inside the target organs. And accordingly, the first neuron is longer and the second neuron is shorter, which is in contrast to the sympathetic nervous system. First, let's look at the sympathetic nervous system. This system is activated in stress, either physical stress, such as exercise, or emotional stress, as well as in frightening conditions, especially in face of a danger. This system induces fight or flight responses, as shown here. These responses involve vigorous body movements, either fight or running away, by the vigorous contractions of the skeletal muscles in the human body. We sometimes refer to the triggers for this system as 3E, exercise, excitement, and the emotion. During the sympathetic nervous system activation, first in the eyes, the pupil dilates so that more light can enter the eye and the person can see better during a fight or during an escape from a danger. Also, the brain becomes more alert for quicker reactions during these responses. And the heart beats harder and faster so that more oxygen and nutrients can be circulated to the contracting skeletal muscles through the blood. And the blood vessels in the contracting skeletal muscle will dilate so that more blood carrying the oxygen and the nutrients can be delivered to the skeletal muscles to meet the higher energy requirement of the muscle during the responses. In contrast, the digestive tract or the GI tract becomes less active during this activation because the activities of this tract is not required during the fight or flight responses. During the sympathetic activation, the lung airways, including the trachea, bronchi, and also the bronchioles, they will dilate so that more oxygen can enter the lungs and to enter the blood. In the meantime, the stored energy in the liver, skeletal muscles, and fat tissues in the form of glycogen and fat, they will break down into small molecules such as glucose and the fatty acids that are released to the blood to provide more energy for the contracting muscles. During the sympathetic activation, the sweat glands are also stimulated. This will help with the heat loss from the skin, which is important for the body temperature regulation, for example, during exercise. In contrast to the fight or flight responses of the sympathetic nervous system, activation of the parasympathetic nervous system triggers the rest and digest responses, such as digestion, defecation, and diuresis, which collectively are called 3D. During the activation of the parasympathetic nervous system, in the eyes, the pupil constricts so that less light can enter the eye, so that the person can have a good rest during this activation. And the lung airways also constrict so that less oxygen is taken in, because at rest, the human body uses less oxygen. And the heart beats slower, and less oxygen and nutrients are circulated throughout the body. In contrast, the digestive tract or the GI tract, as shown here, they secrete digestive enzymes and contract vigorously to promote the digestion of food, to increase the nutrient absorption and to facilitate the storage of the energy in the body during the parasympathetic activation. At certain times, the defecation, urination, or the dilation of genital arteries can occur when this system is activated. To compare the sympathetic and the parasympathetic systems, the sympathetic system causes diffuse effects. This is because each preganglionic fiber connects with many post-ganglionic fibers, so that when the system is activated, it causes a widespread activation throughout the human body. And this system is activated by stress, and it causes the fight or flight response, including increased heart rate, increased blood pressure, and increased blood glucose level. The blood flow is redirected from the skin and from the visceral regions to the skeletal muscles. This is by the vasodilation of the arteries in the skeletal muscle. The purpose of this redirection of the blood is that the contraction of the skeletal muscle is the most important activity during the fight or flight response. In contrast, the parasympathetic nervous system causes discrete or localized effects. This is because each preganglionic fiber connects with only a few post-ganglionic fibers. For example, in a person, when the parasympathetic nervous system is activated, it can selectively reduce the heart rate without stimulating the GI tract activities. The activation of this system is associated with the energy conservation in the body. For example, it causes a decreased heart rate and decreased blood pressure, but it increases the GI tract activity and secretion, and both of these promote the energy conservation in the body. This system also gives permission to empty the bladder, and also the permission to open the GI tract sphincters. In most of the tissues, these two systems oppose each other's actions. The synapse is the structure that connects two neuronal cells, as shown here by this part. Synapse is found in both the central nervous system and in the peripheral nervous system. Here is the presynaptic neuron, and here is the post-synaptic neuron. The second cell can also be a target cell, like a smooth muscle cell, heart cell, or the gland cell. And there is a small gap between these two cells, and this gap is called the cleft, or the synaptic cleft. The major function of the synapse is the signal conduction. For example, when the first neuron gets excited, the action potential is conducted from the cell body to the nerve terminus, as shown here. And this will trigger the release of the neurotransmitters, which are small molecules, and they are chemical signals to be released to the cleft. And these neurotransmitters bound to the receptors, which are located on the membrane of the post-synaptic cell, to trigger the signals in the second cell, so that the signal conduction can be completed through this gap. The major function of the presynaptic neuron is the synthesis, storage, and release of the neurotransmitters into this cleft when the first cell is excited. And the post-synaptic neuron, or target cell, they express the receptors on the cell membrane So they can respond to this, the presence of the neurotransmitters. And when the transmitters bound to the receptors, they will regulate the activity of the second cell for the regulation of the function of the second cell. Here we can see the autonomic nervous system consists of the chains of two neurons, called two nerve fibers that are connected by the synapse in the ganglia. Here is the first neuron. Is called a preganglionic neuron, and the cell body is located in the central nervous system, either in the brain or in the spinal cord. And the fiber of this neuron reaches out to form the synapse with the second neuron in the ganglia. When the first cell is excited, it releases neurotransmitter, which activate the receptors on the cell body of the second neuron. And when the second neuron is excited, it releases neurotransmitter to the target cell. And there are also receptors expressed on the membrane of the target cell so that it can regulate the activity of the target tissue. Again, let's look at this picture in the sympathetic system. These are the ganglia, where the first neuron and the second neuron connect by the synapse. And here are the ganglia in the parasympathetic system, where the first and second neuron connect. Here shows the comparison of the somatic nervous system and the autonomic nervous system. First, we can see the cell bodies. The cell bodies of the first neurons are always located in the central nervous system. In the somatic nervous system, the fiber of the neuron directly connect with the target cell. In this case, is the skeletal muscle, and they form the synapse. When the somatic neuron is excited, the action potential is conducted from the cell body along the fiber to the nerve terminus, and they will release the acetylcholine. Acetylcholine will trigger the contraction of the skeletal muscle. In the sympathetic nervous system, the first neuron reaches out to form the synapse in the ganglion with the second neuron, and this neuron releases acetylcholine to trigger the activation of the second neuron. And the second neuron releases norepinephrine, which will regulate the activities of the target tissues, including the smooth muscle, the glands, and the heart. The nerves of the sympathetic nervous system also directly innervate the adrenal medulla. And when they release the acetylcholine, acetylcholine will stimulate the secretion of epinephrine and the norepinephrine from the adrenal medulla into the blood, and which will also regulate the different target tissues. In the parasympathetic nervous system, both the first neuron and the second neuron use the acetylcholine as the neurotransmitter. And when the acetylcholine binds to the receptors on the target cell, they will regulate their activities. Again, here we can see that in the sympathetic nervous system, the second neuron is longer, but in the parasympathetic system, the first neuron is longer than the second one. Here we can see the neurotransmitters used for the synapse between the post-ganglionic fiber and the target organs. Here, the green color indicates the sympathetic nervous system. In this system, the norepinephrine is the neurotransmitter for the majority of the tissues that are innervated by the sympathetic nervous system, with the only exception of the sweat glands, which are also innervated by the sympathetic system, but they use the acetylcholine as the neurotransmitter. In contrast, the parasympathetic system always use the acetylcholine to regulate the activities of the target tissues. Here is the summary for the acetylcholine, which is the neurotransmitter for first, all the neurons originated in the CNS. For example, the preganglionic neurons for this parasympathetic system, and also for this sympathetic system. It is also used by the sympathetic system that innervate the adrenal medulla. It is also used by the somatic nerve, as shown here. The somatic nerve directly connects with the skeletal muscle, and it also uses acetylcholine to regulate the skeletal muscle contraction. Acetylcholine is also used by the parasympathetic system at the synapse between the post-ganglionic neuron and the target tissues. It is also used by the sweat gland regulation, as shown here. Acetylcholine is also used by some neurons in the central nervous system. There's a question. Are the sympathetic system using acetylcholine in the preganglionic fibers? Yes. All the preganglionic fibers use the acetylcholine as the neurotransmitter, as shown here. Yeah, thank you for the question. The receptors for the acetylcholine are called cholinergic receptors. There are two types of the cholinergic receptors based on their ability to bind to either the muscarine or nicotine. Muscarine is a plant alkaloid found in certain mushrooms, and the nicotine is contained in the tobacco plants, as shown here. The receptors that are able to bind to muscarine are called the muscarinic receptors, or the M receptors. In this group, the M1 subtype is found in some neurons in the CNS and also the autonomic ganglia. The M2 subtype is expressed in the heart, and the M3 subtype is expressed in the smooth muscle and in the glands. The receptors that can bind to this nicotine are called nicotinic receptors, or called N receptors. These receptors are found in the autonomic ganglia and also in the skeletal muscle, in the neuromuscular junctions. And these N receptors can also be found in some neurons in the central nervous system. The acetylcholine and muscarine, these two are the agonists for the M receptors, which mean that when these two bind to the receptor, they will activate the receptor. And atropine is an antagonist for the receptor. When atropine binds to the receptor, it will inhibit the receptor activity. In the mammals, there are a total of 5 subtypes of the M receptors. The M1, M3, and M5 are the first group, and M3 is most important because it is expressed in the smooth muscle and the glands. In this group, when the agonist, either acetylcholine or the muscarine, binds to the receptor, it will activate G protein called GQ. And the GQ protein will activate the enzyme called PLC, phospholipase C, which generates the IP3. It's a small molecule in the cell. And when the IP3 level in the cell is increased, it binds to IP3 receptors on the membrane of the SR. SR means sarcoplasmic reticulum. And SR, the major function of SR is to store calcium in the lumen of the SR. And when the IP3 binds to its receptors on the SR membrane, it will causes the release of the calcium from the lumen of the SR to the cytosol of the cell, causing increased calcium level. In the smooth muscle, for example, in the airway, increased calcium will cause a contraction of the cells, which we'll see the details in next slide. In the blood vessel, when the M3 receptor is activated, the calcium release from the SR will increase the nitric oxide, or NO, production from the endothelial cells. This will cause vascular smooth muscle relaxation, causing vasodilation. In the gland tissues, when the calcium level in the cell is increased, it will stimulate the glandular secretion in the gland tissues. These are the first group. The second group includes M2 and M4. M2 is the subtype expressed in the heart. This group, as shown here, these receptors are coupled with another G protein called GI. I here means inhibitory. Normally, the enzyme AC, which means adenylyl cyclase, AC produces CAMP using the ATP as the substrate. And when the CAMP level is increased, it activates the enzyme pKa, protein kinase A, and pKa will phosphorylate the calcium channels on the cell membrane to increase the calcium influx from the outside to the inside of the cell. In the heart, in the cardiac atrial cell, when the M2 receptor is activated, it will activate the GI protein, and the GI will inhibit this pathway. Therefore, the calcium influx through the calcium channels will be reduced, causing reduced contractility of the atrial muscle tissues. In the heart, in the pacemaker tissue, which is the sinoatrial node of the heart, when this M2 receptor is activated, the beta gamma subunit of the GI protein can directly activate a type of ion channels, which is a potassium channel. And when the channel is activated, it will cause the cell membrane hyperpolarization to slow down the heart rate. Therefore, the M2 receptor in the heart is inhibitory. In the atrial muscle, it will reduce the contractility. And in the pacemaker tissue of the heart, it will lead to slower heart rate. I see there's another question. N receptor bound nicotine and acetylcholine. Yes, that's correct. Acetylcholine can bind to either M receptor or the N receptor. Acetylcholine can bind to both. Nicotine can only bind to the N receptors. Yeah, thank you. Here shows a relaxed single smooth muscle, as shown here. And here shows a contracted smooth muscle. And here is the diagram of the smooth muscle. For the contraction of the cell, it is initiated by the calcium increases, which means the free calcium in the cytosol of the cell. Typically, it is increased from a resting level of 0.1 to 10 micromolar, which is 100 times increase. And this increase can be either caused by the opening of the L-type calcium channel on the cell membrane. When the channel is open, the calcium enters the cell from the outside. It can also be caused by the release of the calcium from the SR. For example, when IP3 level is increased, the IP3 will induce the release of the calcium from the SR. In the second step, the calcium binds to a protein in the cell called the calmodulin. And the calcium calmodulin complex will stimulate an enzyme called MLCK. The full name is myosin light chain kinase. When this enzyme is activated, it phosphorylates the myosin light chain, as shown here, this small white part, by adding a phosphate group, as shown here. When the myosin is phosphorylated, it will be activated, and it can bind to the actin filament to form the cross bridge to trigger the contraction of the cell. So the relaxed cell will start to contract and become this. For the relaxation of the cell, which is the contracted cell return to the relaxed state, the first mechanism is the calcium decreases, which can be achieved either by the inhibition or closing of the L-type calcium channel, or the reduction of the IP3 level in the cell. When the calcium level is reduced, this stimulatory mechanism will be stopped so that the cell can slowly stop the contraction and slowly return to the relaxed state. Another mechanism for the relaxation of the cell is the activation of the myosin light chain phosphatase, as shown here, the red arrow. Phosphatase is an enzyme that removes the phosphate from the substrate. In this case, when this phosphatase enzyme is activated, it removes the phosphate from the myosin to counteract the effect of the MLCK. And when the phosphate is removed, it will also stop this contraction mechanism so that the cell can relax. Any questions? Yeah. Okay, great. Let's continue. Here in the bottom shows the structure of a typical artery. Here we can see artery has 3 layers. The inner layer is a thin layer of the endothelial cells, and the middle layer contains the smooth muscle cells, as shown here. And the outer layer is mostly the collagen fibers. There are very few cells in the outer layer. The nitric oxide, or the NO, is a potent vasodilator, which means it can relax the smooth muscle and causing the dilation of the vessel. NO is produced in the endothelial cells using the L-arginine as the substrate. And this reaction is catalyzed by this enzyme called NOS. And the full name is called nitric oxide synthase, or NOS here. In resting blood vessels, there is a basal level of NO generation. But this generation can be further stimulated by the acetylcholine after it binds to this receptor, the M3 receptors located on the cell membrane of the endothelial cells. This will stimulate the activity of the NOS enzyme to increase the rate of the NO generation. When NO is produced in the endothelial cells, it rapidly diffuses to the smooth muscle cells. And inside the smooth muscle cell, it activates the GC, which is the granulocyclase, to produce cGMP. And the cGMP can cause vasodilation by 3 different mechanisms. The first is the inhibition of the L-type calcium channels. And second is the it can decrease the activity of the MLCK. And thirdly, it increases the myosin light chain phosphatase. Let's go back here. So cGMP, the signal can first inhibit the L-type calcium channel to reduce the calcium level. It can also reduce the activity of the MLCK, and it can also stimulate the activity of the phosphatase. All these 3 mechanisms promote the relaxation of the smooth muscle. Therefore, nitric oxide can cause a potent relaxation of the blood vessels. Here shows the mechanisms of the regulation of the ion channels by the G protein coupled receptors. So ion channels are they are proteins on the cell membrane. There is a pore in the center of the protein. When the channel is activated, the pore will be open to allow the flux of the different ions. For example, the calcium channel allows the flux of the calcium, and the potassium channel allows the flux of the potassium ions across the cell membrane. In the first mechanism, which is in the case of GS, S means the stimulatory G protein. When the neurotransmitter binds to the receptor from the outside, it causes the conformational changes in the protein, which activates the G protein in the inside of the cell. And the G protein is composed of 3 subunits, alpha, beta, gamma. In the case of GS, the alpha subunit will activate an enzyme. In this case is the AC acetyl cyclase, which produces CAMP. And CAMP activates the protein kinase A. And the protein kinase A will phosphorylate the ion channels, causing channel opening to increase the opening chances of the target channel, as shown here. This mechanism is found by the regulation of the calcium channel, as we just discussed. In the second mechanism, which is found in the GI protein, as we said, I means inhibitory. In this case, the beta gamma subunits can directly interact with the target ion channel, leading to the opening of the channel. This mechanism is found by the acetylcholine sensitive potassium channels, for example, in the pacemaker tissue of the heart. When the acetylcholine binds to the receptor, the beta gamma subunits of the GI protein can directly activate the potassium channel, causing the opening of the channel. So these are the 2 different mechanisms for the regulation of the ion channels by the G protein coupled receptors. We have talked about the M receptors. Now let's look at the N receptors. The N receptors are found in the autonomic ganglia. They are expressed on the cell membrane of the post-ganglionic neuron on the cell body. When the acetylcholine is released from the first neuron, the acetylcholine activates these receptors, causing the excitation of the second neuron. The N receptors are also expressed at the junctions between the somatic nerve and the skeletal muscle in these neuromuscular junctions, as shown here. These receptors are ion channels, so they are ligand-gated sodium channels. They are pentamers. Pentamers means they have 5 subunits, as shown here: 1, 2, 3, 4, 5. And in this case, the ligand is the acetylcholine. When the acetylcholine molecules bind to this receptor, as shown by these blue colors, the pore in the center of the channel will be open, which will allow the influx of the sodium from the outside to the inside. This sodium influx will cause cell membrane depolarization, which is also called excitation of the cell membrane. In this post-ganglionic neuron, this excitation will cause the activation of this neuron to cause the release of the neurotransmitters at this synapse to regulate the target cell activity. In the skeletal muscle, this cell membrane activation will cause the calcium release from the SR, which will cause the contraction of the skeletal muscle. These receptors are also found in some neurons in the CNS. They also cause the excitation of the neurons when these receptors are activated. The N receptors in the post-ganglionic neurons, as shown here, these receptors are called NN type, and in the skeletal muscle, these receptors are called NM type, and they have a similar function, but they have different combinations of the subunits. I see there's a question. Do GS and GI have opposite effect on calcium channels? Yes. Let's go back to some slides. Okay, here. Here we can see the AC enzyme can be regulated by either the GS or the GI. So when the GS is activated, it stimulates the AC activity to increase CMP, to increase the calcium influx. But when the GI is activated by the receptors, it inhibits this pathway to reduce the calcium influx. So GS and GI in this case have the opposite effects. Yeah, thank you for the question.
I'll set another question with that one.
Yeah.
It's because on the other slide, it opens potassium channels though, right? The GI?
Yeah.
Okay.
Oh, yeah. So they are in different tissues. If is in the atrial muscle, this special channel is not expressed. This special channel is only expressed in the pacemaker tissue. So in the atrial tissue, they cannot affect this potassium channel because this specific potassium channel is not present on the cell membrane. Yeah, thank you. So here's the summary for the cholinergic receptors. The M receptors are G protein coupled receptors. The M2 reduces the heart rate and reduces the heart muscle contractility. And the M3 receptors stimulate the smooth muscle contraction. It causes vasodilation, and it also stimulates the glandular secretion in the glands. The N receptors are ligand-gated sodium channels. In these post-ganglionic nerves, they cause the excitation of these neurons. And in the skeletal muscle, when the N receptors are activated, they cause the calcium release from the SR to trigger the contraction of the skeletal muscle. Now let's look at the receptors for the norepinephrine and the epinephrine, which are called the adrenergic receptors. These receptors are further divided into alpha 1, alpha 2, beta 1, and beta 2. The alpha 1 receptor, as shown here, this receptor is located in the cell membrane of the smooth muscle, for example, in blood vessel, in the urinary bladder, and also in the eyes, in the iris muscle. The receptor is also expressed in the exocrine glands and in some neurons found in the CNS. And this alpha 1 receptor is also coupled with this GQ, PLC, the enzyme, to produce IP3 to cause the calcium release from the SR. In the smooth muscle, this will cause contraction, and in the glands, alpha 1 receptor will stimulate the secretion from the glands. Alpha 2 receptors, as shown here, these receptors are expressed in these presynaptic neurons, as shown here. When the norepinephrine, NE, is released from this nerve to the cleft, in addition to stimulating the alpha 1 receptor on the target cell, this NE can also bound to and activate the alpha 2 receptors in this presynaptic neuron. This will reduce the CAMP, reduce the calcium level. This will inhibit further release of the NE to the cleft. So it's like a negative feedback mechanism. These receptors are often called autoreceptors. The alpha 2 receptors are also found in the blood platelets. When the alpha 2 is activated, the CAMP level will be reduced in the platelets, and this will promote the platelet aggregation. The beta receptors include beta 1 and beta 2. Both receptors are coupled with the GS, and the GS, as we learned, GS stimulates the AC to increase CAMP, increase PKA, to increase the calcium influx. In the case of the beta 1, which is expressed in the heart and in the kidney tissues, the GS, as shown here, will increase CAMP, increase the calcium influx. In the heart, this will increase the heart rate and increase the contractility of the cardiac muscle. In the kidneys, this will stimulate the secretion of the renin from the kidney cells to the blood. Beta 2's receptors are expressed on the smooth muscle. They are also coupled with AC to increase CAMP. But in the smooth muscle, the CAMP will inhibit this enzyme, as shown here. The minus sign means inhibits. It inhibits the MLCK. Because MLCK promotes the contraction by phosphorylating the myosin. Inhibition of the MLCK will cause relaxation of the cell. In the blood vessel, it will cause vasodilation. Here shows the autonomic control of the pupil size. which is regulated by the contraction of the iris muscle. Here we can see there are two layers of the iris muscle. The inner layer is called the circular or the sphincter muscle. And this muscle express the M3 receptors for the acetylcholine. During parasympathetic nervous system activation, acetylcholine is released, which activates the M3 receptors in this muscle. By the IP3 pathway, it will cause the contraction of this muscle, causing pupil constriction, which is called meiosis. In the outer layer of the iris muscle, which is called radial or the dilator muscle, this muscle expresses the alpha-1 receptors for the norepinephrine. During the sympathetic activation, the norepinephrine is released to this muscle, and it will activate the alpha-1 receptor. Interestingly, by the same IP3 pathway, it will cause the contraction of the outer layer of the muscle. So the pupil will dilate, as shown here, which is called mydriasis. Okay, here is the last slide for this lecture. Here shows the major steps for the synaptic transmission. Here is the presynaptic neuron, and here is the post-synaptic cell. In the first step, the neurotransmitter is synthesized from precursor inside the presynaptic neuron. And once the transmitter is synthesized, it is transported into the lumen of the vesicles, and they are stored here. In the second step, the action potential is generated in the cell body of the neuron, and is conducted through the cell membrane to the cell terminus. And when the action potential reaches this region, it will cause the cell membrane depolarization, which will activate and open the calcium channel. When the calcium channel is open, the calcium will influx from the outside to the inside. And to increase the calcium level inside the cell, this will cause the fusion of these vesicles with the plasma membrane, so that the neurotransmitters can be released to this cleft. In next step, when the neurotransmitter concentration in this region is increased, they bound to the receptors, which are located on the membrane of the post-synaptic cell. They can either open the ion channel or activate the G protein coupled receptors to regulate the activities of the target cell. In this step, the released neurotransmitters can be degraded by the enzymes so that they are destroyed. And in the meantime, some of these neurotransmitters can be transported by this transporter protein located on the presynaptic neuron cell membrane to back to this presynaptic neuron so that they can be recycled. When the neurotransmitter is either degraded or they are transported back into the cell, the level of here will be reduced again. Remember, the bonding of the neurotransmitter to the receptors is reversible, which means when the level of the neurotransmitter is reduced, they will dissociate from the receptors so that the stimulation of the receptors will be stopped and the cell will return to its resting status. That's all the slides for this lecture.